Simsen Diagnostics is a company that is probably one of the best in the world to find extremely small fragments of DNA. And this is based on a technology called the Simsen-Seq, which was developed by professors Tony Godfrey and Anders Ståhlberg at Boston University and Sahlgrenska, respectively, back in 2017. Based on that technology, the company was founded then 5 years ago. 2 years ago, we moved to the BioVenture Hub here at AstraZeneca. And the main application for being able then to make some use of, we can find small fragments of DNA. So what? Well, for example, ctDNA, circulating tumor DNA, is today the main application. We've also entered into yet another application. Sorry, there we go. Which I'll come to a bit later. ctDNA, that's circulating tumor DNA used in cancer monitoring and clinical trials for oncology drugs. So basically what it is that all cells in the body die eventually, and when a tumor cell dies, it sheds off small fragments of the tumor into the bloodstream. That's what we call circulating tumor DNA. This ctDNA, when passing through the bloodstream and the various organs, dissolves within a matter of hours. Meaning that if we can find ctDNA in the blood, then we know there is a tumor present right now, not a week ago, but right now, really. So through something called liquid biopsy, we take blood and we analyze the blood to look for the ctDNA. What makes our approach so sensitive is that we use the actual individual tumor to determine what sort of mutations are we really looking for. So instead of having a tumor agnostic panel, where you basically look for the most common mutations in breast cancer, prostate cancer, whatever, we take a biopsy sample and we analyze that so we know exactly what are the mutations your individual tumor has, and then we know what to look for, meaning that we get an extremely high sensitivity and specificity. The technology is used after surgery because we need the tumor biopsy. And then we can see the treatment. Does it work? Does it affect the tumor burden somehow? And then usually patients are monitored for years and years afterwards. And then we can see also, is there a recurrence somewhere along the line? For example, we work with the Sahlgrenska University Hospital, the pediatric unit, where now all pediatric patients are monitored with ctDNA analysis. We develop the panels, they do the actual analysis themselves. And we've been able to discover recurrences, in this case 9 months prior to clinical presentation, that is when it's visible on imaging. So you get a heads up, very, very fast. We also work primarily, I would say, with several pharmaceutical companies and their clinical trials. Finnish, Swedish, other companies. We have an ongoing relationship with a CRO based in the UK called Theradex Oncology. So they can offer our services to their customers. So that's also a way to the market. And in addition, we have various researchers that we also work with in the medical field. We've recently developed something we call the Simsen's Lab Suite. So essentially we have 2 ways of bringing our business to the client. One way is the full scope, which is what pharma companies normally would use, where we get the biopsy, we analyze it, we get the blood samples, we provide a report back on the ctDNA levels, and they can get an immediate feedback on treatment efficacy. The other way is, and primarily in Europe, hospitals are very reluctant to outsource all sort of analysis to external vendors. So like we do for Sahlgrenska, we also want to get into other hospitals now with this offer where we develop the panels and then we do a know-how and tech transfer to these hospitals so they can do the ctDNA analysis themselves. So they have then complete control of time and costs of everything they do. And we think that's a very attractive offer that we're now in negotiations with a number of other hospitals to see if they want to try this out. apart from just the monitoring part, being able to spot recurrences much earlier than with standard of care, it can also inform treatment. So there was this study, I think 1-2 years ago it was published, where you had two arms. One had ctDNA informed treatment, the other one had standard of care. So basically, for example, colon cancer, you have a 6-week window after which, 6 weeks after the surgery, you need to come to a decision whether to use chemo or not. And that's based on a number of factors. So this study had two arms. One had ctDNA as one parameter of that decision. If there was no or very little ctDNA, meaning no or very little left of the tumor, there was no chemo. If it was high, there was chemo. And then the other arm did not have ctDNA as one of the parameters for decision. The result was equal survivability. There was no change in survival. However, in the non-informed arm, it was 28% of all patients that received chemo, whereas only 15% received chemo in the ctDNA informed. So there was an overprescription of chemo, really. And the cost of chemo is not very high. The costs have actually gone down quite a lot for those drugs. But if you look at all the side effects, peripheral neuropathy, quality of life indicators, etc., there's a lot of cost savings that can be done by reducing chemo. This is not an argument we use to get into the clinical trials where people want to sell more drugs. But to the healthcare system, there's a lot of money to be saved by using ctDNA to inform treatment decisions. Now, we're very, very good, as I said, about finding these small fragments of DNA. So 2 years ago, there was a project in which we and the NFC, the National Forensic Center, were involved. And we were discussing and developing a small sort of prototype panel for forensic use to be able to more exactly and more sensitive than today to determine whether a DNA sample found on a crime scene could match any database or a number of suspects or whatever. And we developed a panel actually in that project that was pretty outstanding, actually surpassed everything that was on the market. But then nothing was really done. So close to a year ago, we reinitiated the discussions with NFC, and now we've started developing a forensic panel. Really, a lot of these cases are never solved because of the sensitivity being way too low in the existing panels. So that's what we hope to accomplish now. And you have all of these cases with nowadays war or mass disaster victims and everything else where you need to positively identify the various victims. Sensitivity is not on the level where it could be until now. So with the Sekifox, which is the working name at least now for this new product, we intend to help solving lots and lots of more cold cases, working with forensics globally. This is outside of the medical regulatory framework, so we don't really need to seek the same sort of approvals. We need to work with the forensic labs, we need to validate according to their standards, and the Swedish NFC has put up a number of standards, and together with them we now have a validation protocol that we are pursuing, and we hope to be able to commercialize this early next year because validation will be done November, December this year. So as I said, we had 7 so-called STR markers that we had 2 years ago that we tried out, and in a published paper it was found to surpass the effect of many of the existing commercial alternatives. Now we're evaluating a 23 markers set, and that will be much more sensitive. And according to NFC, there is nothing on the market today with that sensitivity. So they are very eager for us now to finalize the validation and start commercializing it. And then we'll have an international launch next year. We've already actually been in touch with a number of primarily European forensic laboratories And only this morning, the Italian Carabinieri sent a mail that they now want to meet and discuss this because there's a lot of interest. There's so much now that can be solved that couldn't be and can't be today. So yes, on one hand, we're now into forensics. On the other, we're in the medical, but it's all based on the same technology, the same R&D resources, the same value proposition that we now just explain how that is relevant to our different target groups. So we're not really diversifying completely, doing something rather utter different, but really we're staying with the technology, just finding a new application for that technology. But we need money, and that's also a reason why we now ventured into forensics, to balance the risk, because our clients within clinical trials, they're small, medium-sized pharma companies, they're struggling. They're trying to find financing to run their trials. And we see a lot of trials today being delayed or cut down, or companies going bankrupt, and we're a bit lower down in the food chain, so of course that affects us. We still have a number of really good large clients, many clients that we're working with in ongoing trials right now, but we want to balance the risk somehow, because when we're personalized within the medical space, here we develop one kit which we can manufacture 1 or 1,000 or 10,000, doesn't really matter, it's just one and the same kit used in forensics. So that's very transactional and fairly low risk, so to speak. But to sustain the business, we're now looking to raise 20 million Swedish, 2 million euros approximately. We hope to close it by the end of the year. The money will be used to sustain the business, it will be used to grow internationally, it will be used to launch Sekifox internationally, and putting more R&D efforts into both the oncology to further enhance the technology and the bioinformatics software that we use, but also work towards a CE marking for the medical application. And we're a bit in the dark as to the total cost of that. We're in discussion with TÜV Syd to better define the need for clinical studies and to what extent we can claim parity with existing literature. Thank you so much. Questions, please.
Thank you very much, and please raise your hand if you have any questions. I start off. Interesting, you have this dual business, so to say. And which do you think will be the big one in the long term? Is it the forensics that has the biggest potential, or is it the medical part?
Right now we're generating money from the medical application, oncology, primarily through clinical studies. And had we spoken a year ago, I would have said by far it's within oncology. But again, the financial situation and their ability to finance the future studies is causing a lot of delays. So short term, I think we'll see forensics kick off fairly fast. But long term, I guess, sometime, we all hear about the uncertainty right now. I don't think that there is time to wait for things to be more certain. I think the uncertainty is the new reality. We need to learn how to navigate that. So when people wait to be more certain, that's not going to happen for at least 5-6 years. But in 5-6 years, the studies and with a CE mark at that time, also the ability to implement the technology and the solution into hospitals, that will cause a lot of recurring business.
And the main customer on the medical side, is that a CRO or what kind of end user do you have at the moment?
Currently it's the pharma companies themselves.
Okay.
And the deal we have with the CROs that we're working with is that they take it as a pass-through cost, meaning that they strengthen their offer, that they can offer ctDNA analysis to their clients, but we would charge them as much as if we were charging the end customer directly, and it would just be a pass-through cost. That's not how the CRO makes money. They make money on the planning and driving and project management.
Of course. And the owner, who owns the company at the moment?
Well, the cap table is unfortunately not VC fit, meaning that the people that work in the company don't own as much. However, the owners are the founders, the two founders, the professors, Anders Stålberg and Tony Godfrey, together with a smaller piece from GU Ventures. There's a family office and a number of angel investors.
Okay. Thank you very much, Pontus. We have to move on.
Sure. Thank you.